Welcome to This Explainer. Today, we're going to trace what is honestly one of the most amazing journeys in modern science. We're talking about the incredible pipeline that takes microscopic organisms, sometimes literally pulled straight from the dirt, and transforms them into the everyday industrial products and life-saving drugs that we all rely on. It's this fascinating, high-stakes mix of biology, engineering, and just absolute sheer scale. So you might be wondering, how exactly do we turn a regular old handful of dirt into life-saving medicine? I mean, it sounds a bit like alchemy, right? But it's actually the very foundation of modern bioprocessing. The mundane soil right beneath our feet is just teeming with microscopic workers, and figuring out how to harness them is what makes massive industrial cures a reality. Here is our roadmap for today. First, we'll cover the bioprocessing pipeline. Then, hunting for microscopic factories. Next up is microbial versus mammalian production, followed by purification and packaging, and we'll wrap up with scaling up and economics.
Let's kick things off with Section 1, the bioprocessing pipeline.
You can really think of industrial biochemistry as the ultimate manufacturing plant. But instead of heavy machinery stamping out pieces of steel, we're applying biological systems. We use enzymes, microorganisms, and cells to produce useful products on a massive, highly controlled scale. In this analogy, the cells are our tireless little workers, and the massive stainless steel bioreactors, those are our highly optimized assembly lines. And every single product built this way follows 3 universal steps. First step is upstream processing. This involves preparing the media and optimizing the growth conditions so our cells are, well, happy. Second is fermentation. This is the core production phase where the cells actually synthesize the product. And finally, downstream processing, where we separate, purify, and formulate the final product so it's completely safe to use. This pipeline is basically the absolute blueprint for everything we're going to discuss today. Moving right along to Section 2, hunting for microscopic factories. Now, before we can even think about scaling anything up, we actually have to find these microscopic workers out in the wild. Imagine a microscopic battlefield. This right here is the crowded plate technique. When scientists take a soil sample, they plate a whole mixed population of microbes all together. If a colony, say a Streptomyces species, naturally produces an antibiotic, it actively secretes it outward. This literally kills off or halts the growth of competing microbes nearby, creating a clear ring known as the zone of inhibition. We are literally using their natural chemical warfare to find our next generation of drugs. But sometimes we need a very specific worker, so we use an enrichment culture. Instead of just looking at who survives in a crowded space, we create a highly tailored environment. We apply specific nutritional or environmental pressure, like, for example, providing uric acid as the only nitrogen source available. By doing this, we guarantee that only the specific microbe with the exact right metabolic tools can outcompete the rest. It's a super elegant way to force the exact microscopic factory we want to just reveal itself. All right, part 3, microbial versus mammalian production. Once we've found our worker, it's time to step onto the actual factory floor to look at the vastly different types of bioreactors and cells we use for production. With microbes, we generally choose between 2 main fermentation methods. Solid-state fermentation, or SSF, involves growing organisms on solid substrates with very little water. Think wheat bran or rice bran. This is brilliant because it perfectly mimics the natural ecological niche of fungi, making them highly efficient at producing things like industrial enzymes. On the flip side, we have submerged fermentation, or SMF, where cells grow entirely suspended in liquid. While this requires massive, expensive tanks, it allows for incredibly intense and precise control over oxygen, pH, and temperature. But here's the catch. Microbes have a massive limitation. They are terrible at making complex human proteins. So we turn to mammalian cells. Enter CHO cells, which stands for Chinese hamster ovary cells. Yep, you heard that right. It sounds bizarre, I know, but hamster ovaries are the absolute industrial standard. They are responsible for producing over 70% of approved recombinant therapeutic proteins. Why? Because they can actually fold proteins properly and add human-like sugar molecules, a crucial process called glycosylation that bacteria simply cannot do. And this is a brilliant example of exactly why we rely on these hamster cells. We're talking about the production of tissue plasminogen activator, or TPA. It's a highly complex secreted glycoprotein. In a clinical setting, TPA catalyzes the conversion of plasminogen to plasmin, which literally dissolves blood clots. If someone you love is rushed to the hospital actively suffering from a stroke or a heart attack, this is the drug that saves their life.
A simple bacteria would completely misfold this protein, making it useless or even dangerous. CHO cells get it right every time. Watching this build into an industrial process is honestly pretty stunning. We start by inserting the TPA gene into a plasmid, introducing that into the mammalian CHO cells, and growing them in a tiny petri dish. From there, we scale way up, transferring those actively secreting mammalian cells into massive industrial fermenters. The cells do all the heavy lifting, actively secreting the life-saving TPA directly into the liquid culture medium for us to harvest.
Next up is Section 4, Purification and Packaging. So your product is now just floating in this liquid broth. Now what? Well, downstream processing kicks off by completely removing the cells via centrifugation. Then we purify the product using chromatography. But take a close look at the third step there, viral clearance. This is an absolute, non-negotiable regulatory safeguard. Because mammalian cells can naturally harbor viruses, we have to use low pH treatments to destroy viral envelopes, along with nanofiltration to physically catch microscopic viral particles. Only after this strict clearance do we formulate the final product, often freeze-drying it to maximize its shelf life. Now, all of this intense processing raises a really critical question. How on earth do we safely dispose of the massive chemical and biological waste left behind? Because, you can't just dump hundreds of thousands of liters of unused nutrients, discarded cells, and solvent waste straight down the drain.
The answer here is incredibly elegant. The factory literally uses a biological solution to clean up its own biological mess. It's called the activated sludge process. By using specialized aerobic bacteria and actively pumping in oxygen, the factory breaks down organic waste. In places like a penicillin plant, for example, this process degrades dangerous antibiotic residues way before the water is ever released into the environment. This strictly prevents the catastrophic spread of antimicrobial resistance.
Finally, let's look at Section 5, Scaling Up and Economics. Making a drug in a lab is science, but making 1 million doses reliably and affordably, that is pure engineering. The sheer scale of growth over just a couple of weeks is absolutely staggering. You start on day 1 with a tiny 100-milliliter shake flask. By day 5, you've moved the culture up to a 50-liter pilot bioreactor. And by day 15, you are managing a 5,000-liter industrial bioreactor. The engineering challenges involved here are immense. You have to ensure oxygen mass transfer reaches every single cell in that giant tank, all while carefully managing the mechanical shear stress of the impellers so you don't literally tear your delicate mammalian cells apart. And as you might guess, the economics behind this are super intense. You have massive capital expenditure, or CapEx, up front because those massive steel bioreactors and sterilization systems are certainly not cheap. Then you have continuous operational expenditure, or OPEX, paying for perfectly balanced raw materials, immense utility costs for heating and cooling, and of course, a highly skilled labor force. On top of all that, ensuring good manufacturing practices and securing FDA approvals adds a monumental layer of regulatory compliance costs. It is a high-stakes, multi-million-dollar balancing act every single day. So we've seen how a tiny microbe in the dirt can be isolated, engineered, scaled up, and purified to literally dissolve a blood clot in a human heart. It completely redefines what manufacturing actually means. I want to leave you with this final provocative thought to wrap up this explainer. With living cells now acting as our most advanced programmable factories, what on earth will we program them to build next?